Hello, everybody. Welcome to our distinguished talk presentation from the Solid State Circuits and EDS South Brazil Society. Today we have two guests. Actually, our presenter, Dante Muratore, will talk about near sensor compression, high bandwidth brain-computer interfaces. And to host him, we have Dr. Oscar Mattia, who is an IC Brazil program graduate. It's very nice to have him here. Thank you for accepting. And also had a master on microelectronics at URGS and PhD from the Free University of Brussels with honors. Thank you very much for coming. The floor is yours. You can please introduce our guest.
Thank you, Sandro. And it's a pleasure to introduce a former colleague and good friend, Dante Muratore. Dante is an assistant professor of bioelectronics at Delft University of Technology, and he received his bachelor's degree and master's degree in electrical engineering from Politecnico di Torino in 2012 and 2013. He received his PhD degree in microelectronics from University of Pavia in 2017. From 2015 to 2016, he was a visiting scholar at MTL Labs at the Massachusetts Institute of Technology. And from 2016 to 2020, he was a postdoctoral fellow at Stanford University. He's the recipient of the Woodside Neurosciences Institute Interdisciplinary Scholar Award. His research focuses on hardware algorithm co-design for brain-computer interfaces, bioelectronics, sensor interfaces, and machine learning at the edge. Thanks for being with us, Dante, and the floor is yours.
Thank you, Oscar, and thank you, Sandro, for having me here. It's a pleasure to be talking about this topic today. So, as we said, basically, today we're going to be touching on high bandwidth brain-computer interfaces, and specifically why we need to do compression as close to the sensor as possible. Right. So let's start by defining what a brain-computer interface is and what it can do. Right. So as you can imagine, it has a brain and a computer in the loop, and so the basic idea is that we can place electrodes in the brain to record neural activity, and then do something with this, right? And so basically, the concept is that we record this neural activity, we process it, and we decode some sort of information, and then we can use to either control a prosthetic device like a robotic arm or a cursor, or even directly decode speech from these signals. Or we can use these to, for example, control a closed-loop neuromodulation device that also stimulates the cells in the brain, right? And so you have this very nice, beautiful thing that allows you to record and stimulate. And now the question is, well, what can you do with it? And it really depends on the type of signals that you're interested in recording, right? And so you have a fundamental trade-off between the sensor size and the location, and so for how many, if you want to have very small sensors that record the unit of information of the brain, which are these spikes that come from the neurons, you can not cover a very large area of the brain, right? And so you have a trade-off between resolution and the sensing area. So today we're going to be focusing specifically on brain-machine interfaces that can record action potential, so the activity of single neurons. Now, here's an example of what you can do with this type of devices, which I was trying to put up front to show the promise of this technology, right? And so in this case, you see on the top of the head of this patient that suffers from paralysis, the implanted device has 100 channels that can record neural activity, and they record neural activity from the motor cortex and they use that to decode the intention of movement of the patient. So the patient cannot move anymore, but can think of moving. And so with that information, then they can control cursor on a computer and then use that to allow the communication to type on a keyboard and then communicate to others, right? And so this is the first time I saw this, I was blown away, and I guess that's why I'm kind of like working on this field. Is that this is great technology. This is actually quite old. It's by now it's going to be almost 10 years old results. But you can see that while quite impressive, the number of correct characters per minute they can achieve are still very low, right? So the typing speed is nowhere near that of a person without any motor problems. So what is the issue here is that you're trying to decode the intention of movement that is typically encoded in hundreds of millions of neurons by recording only from 100 channels, right? And so clearly there is a need to increase the bandwidth of the communication with the brain, and there is a clear trend that you can observe, which goes towards having more and more parallel recordings over the years, and this you can come out with something similar to a Moore's law, but with the number of simultaneously recorded neurons, with a sad reality that the doubling time is a lot slower, right? So we double the number of electrodes roughly every 6.3 years. But however, the long-term goal is to have full populations of neurons at single-cell resolution, right? So if you want to start recording from more neurons, then you will need to record from more channels, and so you will be generating more data. And the problem, the current bottleneck when you look at this is that of the wireless power transmission, of the wireless data transmission. So the power to transmit this data wirelessly. And so here is from a paper we published a couple of years ago now, that basically studies different systems and tries to compare what is the power for the amplifier, the ADC, and the transmitter based on, let's say, first principle models and state-of-the-art results. And almost in every system, the total power is dominated by the transmitter power. And so this hopefully justifies the need for compression in the implant. So now we have why we want high bandwidth brain-machine interfaces and why we need to compress. So the rest of the talk is gonna focus on compression. And I split the talk in two parts. The first is kind of a tutorial of what is the standard way to approach this, which is what I call signal-driven compression. And then we're gonna look into the approach that my lab is pursuing, which is something that doesn't look so much at the signal specifically, but at the output of the entire processing pipeline. And so what do you want to do with the signal, and how can we take that into account when applying compression? But first, let's look at signal-driven compression so that we know, where we can start from, right? And so we're going to look at three very simple examples. So as I said, signal-driven compression, here the goal is to compress the data and then being able to reconstruct and have as much fidelity as possible, right? So this is kind of like a lossless compression approach. And so if you start from the very most conventional way, is that of compression by transformation. This is your typical JPEG, right? Where you simply say that real life signals are usually sparse in some domain. It doesn't necessarily need to be the time domain in which we typically record. But the idea is that if you have your input, you have some domain. In this case, let's take a DFT of this input on the left. And so we're going to look into the frequency domain. Then in that domain, you should have a sparse representation. Now, what that does mean, it means that in that domain, the entries of the coefficients of your output are mostly zero, right? And so in this case, the signal on the left, you will need to sample a Nyquist. In reality, it's just a toy example that I did on MATLAB, which is a sum of a very few sine waves. And so you do not need to store all the values, but only the coefficients that are non-zero, right? And so this is the very basic approach. So you transform to the domain where you have sparsity, and then you compress. Now, why can't we do that in brain-machine interfaces? Is that if you think about the pipeline, how it would look like is that you have your analog front end with your ADC, and then you would need to have all the samples at Nyquist, so that then you can move to whatever domain you want to move to, and then you can do the compression only afterwards, right? And when you want to have thousands or tens or 100,000 channels, then generating all this data is actually quite power hungry, and so it's something that you cannot afford. And we're going to look at a very specific example in the retina for this, where we have some numbers. For now, you will have to trust me that this is just not going to fly when you want to go for many channels. So around 2005 or 2006, some people came up with this very cool idea of compressed sensing, where basically solves, in a sense, solves this problem that says you don't need to store all the samples before you do your compression. And so if you have your time domain signal, you can design a sampling matrix that it doesn't necessarily need to be diagonal, and get sub-Nyquist samples of this time domain signal. And if the signal is sparse, then you should be able to reconstruct without many errors. Now, if the signal is sparse, it's a key concept that you need to keep in mind, right? Because if it's not sparse, if it's wideband, then, well, Nyquist was not wrong, right? So then you would have aliasing, and this would be a problem, right? And so the concept is that you can represent X as a sparse signal in some domain. And so then you want to solve for S. And this is, if you just try to solve for this, then you have an underdetermined problem, which is basically you will have aliasing. There are many S's that will satisfy Y. And so what you want to find is the sparsest possible S. And the key innovation here is that typically to find sparsity, you will need to have the L0 norm. That's a very hard problem to solve. And so instead, you can show mathematically that even if you use the L1 norm, which makes it something that you can solve much easily, then numerically, then this still works with minimum error, right? And so the nice thing is that now you don't need to sample all the Nyquist samples, but you can just get the compression during the sampling itself. Of course, you have some caveats, so the signal must be sparse in some transform, but luckily for us, almost all real data is, and this is because real data is structured data, and so then that kind of like guarantees sparsity. Then your measurements must be incoherent, and this puts some constraints on your sampling matrix. And then you need sufficiently many measurements, which put some constraints on the size. So don't worry, we're not gonna go much into the math, but you need to know the basics to then understand the actual hardware implementations of this and why this actually makes a lot of sense for implantable devices. So basically, one nice trick also is that if your sensing matrix, here called phi, is random, then the basis does not need to be known a priori. And so you can have a very generic front end that doesn't even know anything about the data, and then you can figure out the domain in which, the basis in which your signal is sparse afterwards, right? And so what you look at here is basically you get Y by doing a linear projection of X on this sampling matrix, right? And so this is very simple to implement because a linear projection is just multiply and add, right? And so especially now with the whole boom in machine learning, multiply and adds are kind of multiply and accumulate are kind of like the bread and butter of hardware designers. And so here are two examples from a paper from Shoran where you can do this both in the analog or in the digital domain, right? So now the domain, basically, your multiplies are mixers and your accumulators are integrators, and you can design these sampling matrix such that your coefficients are binary. And so then this mixer becomes a simple switch, which makes it very simple, very easy to implement. Or you can do it in the digital domain. Now, if you do it in the digital domain, you need to capture the data at the Nyquist rate to begin with, right? So this might become a problem if you want to go for a high channel count. And another thing to keep in mind that in the analog domain, you need M channels, where M depends on the size of your sampling matrix, which depends on the level of sparsity of your signal. So you have trade-offs here. This is not perfect, but it's a great way to get compression for a very low cost, because the encoding part, which is where you're actually doing the compression, is very cheap. Then the decoder is actually a nonlinear problem, which makes everything harder, but that can happen outside of the implant, right? And so this is what we say here is that if you have a sensing matrix that is random, which is what you would normally want to do, then your optimization problem is nonlinear and compute intensive. Now, typically, this is not a problem because you can do this outside of the implant, and that's completely fine. But if you want to do anything in real time, then this becomes an issue, right? And so what Aprile and others did a few years ago was introduce something called learning-based compressed sensing, where basically it says, well, if I know my base is a priori, so I need to know something about the signal, that's okay. I mean, typically we do. Then I can learn a sampling matrix. It's not random anymore, but I just learn basically which rows and columns should be non-zero in here. And then my decoder becomes also linear, right? So I still have a linear encoder. This is just a multiplication and accumulation. This is very simple. But also my decoder now is just inverting the matrix. And so this is very nice because I can do real-time operation. I can put all sorts of constraints on this sampling matrix as I'm learning and play very nice tricks, right? And in here on the right, you see a very simple, I don't know why these things zoom in, but okay, there you go. You have a very simple implementation where you basically just can learn these sampling matrix coefficients on the fly, and then you just have a finite state machine that looks at a lookup table for the coefficients, and then you just do your multiply and accumulate in the digital domain, right? And so this is compressed sensing, and just the take homes here to have is that if you don't know your transform basis, then you are kind of like stuck with a nonlinear decoder. But if you know something about the signal, then you can learn your sampling metrics, and then everything becomes linear. And so you can also look at the way you design these sampling metrics on the basis to have very efficient hardware implementation that gives you basically binary coefficients. And then your multiplication becomes an XOR in the digital domain or a switch in the analog domain, right? And if you are more interested, Shoran in 2014 show how to export this to multi-channel. However, these are the problems I was telling you about, that multi-channel compressed sensing is not as area efficient as other solutions, right? So this is to keep in mind, and this is also one of the reasons why it is okay for like low channel count applications, but it becomes very hard for like thousands or hundreds thousand channels applications. So the other approach you can go about is something called autoencoders, which is basically your typical deep neural network where you have an input layer, some hidden layer in the middle, and then an output layer. And so this is probably you heard about this a lot for classification, right? And so basically you have a nonlinear encoder and a linear decoder, and then you achieve compression by transmitting the hidden layer. Now you need to design this such that the dimensionality of the hidden layer is smaller than the dimensionality of the input layer, right? Otherwise, you don't get any compression, but this is something you can do. And then, as any artificial neural network, then basically you want to learn your weights, which are your decoders and encoders, to minimize the input-output Euclidean distance. Now, in here, you want to minimize the input-output because ideally you're trying to reconstruct the input identically, right? This is not a classifier. And so as usual with these deep neural networks, well, the accuracy comes from them being deep, and so you have a trade-off between the size, which turns out to be power and area, and the accuracy. But the nice thing about this is that they can be trained to be quite robust against signal variations, and so they tend to be more robust at, for example, compressed sensing approaches or other approaches. And you can also design them, like in the approach by Wu, to be heavily asymmetric. This is something that you always want to do when you're thinking about implantable devices, right? Because you have a heavily asymmetric system where your implant is constrained in area and power and how much data you can transmit, and basically anything you can think of is constrained in the implant. And outside, it's almost Christmas, right? So you can burn more power, you can have a battery, you don't need to do, you can have wires if you need to, and so on. And so here, for example, the number of parameters that you have on the encoder part is 40 times smaller than the ones on the decoder, right? And this is something you need to keep in mind. Also, you can turn these autoencoders into multipurpose autoencoders, where basically you do the first part, you get to the hidden layer, that's where you transmit after W2, and then on the decoder, you can either reconstruct your signal if you're interested, or you can train for classification, and you can directly decode what is the, for example, the emotional state, whether the patient is awake or not, or directly maybe the velocity vectors for the example I showed you before. Now, if you can bring also the classification inside the implant, then you have ultimate compression, where basically you just send out the information that you need, right? However, now you have a lot of computation to get there, and that might not be the best solution. So this is always a trade-off: how much compression versus the cost of that compression you want to put in. However, in here, the take-homes, you can think of autoencoders as compressed sensing, where you learn your custom transform basis, and you usually need a lot of big data for training that. And then you have this model size versus performance trade-off. But this extra complexity in hardware gives you a robustness against signal variations, right? And then you can do it for reconstruction, which is standard, let's say, signal-driven compression, or classification, which is something I would call output-driven compression. However, this is not, again, very suitable for like large channels, unless you want large channel count, unless you want to do classification on top. But we are still far away from something like this. So this brings us to our approach, where is basically taking a step farther and saying, I don't want to even, I don't want to guarantee that my signal is faithfully reconstructed. What I want to guarantee is that my output is the same as if I don't do any compression, right? So we can have, from the signal point of view, lossless compression, and then lossy compression, and then we would like to have, from the output point of view, lossless compression, right? So I'm going to give you two examples of work that that I did in the past. One is for a motor BCI, and the other is for an artificial retina. So the first one I call low-hanging fruit, and you'll see why in a second. But as I said, the justification for going for output-driven compression is that if you think about your signal frequency, this is your Nyquist rate, let's say, that's much higher than your actual signal information. So this is the concept of your signal being sparse in some domain, and so this is the compression, signal-driven compression step. And then typically your signal information is much higher, the rate is much higher than the application information rate, and this is the concept of you need 100 neurons to encode whether you're moving to the right or to the left, right? So the dimensionality of the input is, or to the motor decoder in this case, is much higher than the dimensionality of the output. And so you should basically do compression based on your lower dimensionality outputs, and that should allow you to hopefully find a better trade-off in compression versus performance. And if you look at the signal, it's gonna get more distorted, but maybe you don't care about it, and that's what I'm gonna show you in the next two examples. So what is brain-computer interface, intracortical brain-computer interface in this case is the video I showed you before, but here is just the breakdown of how this works. You have your microelectrode array that records basically neural activity. These are all these, let me see if I can see. Okay, now you should see the laser. These are all these time versus voltage traces here. Each trace is a different channel. And then you can process these, and then you can do something called spike sorting. This is what SS stands for, where we basically look at the spikes in each of these channels, and then you say whether it comes from neuron A or neuron B. And so in here, each row is not a channel anymore, but it's a single spike. And so spike, the black spikes fire at these times, and the yellow one at these other times, and the magenta, and so on, right? And then you put all of these into a decoder, and then you decode basically the motor control for the processes. So the Y and X velocity vectors in the case of a 2D space, right? And so this project works within the consortium that is called BrainGate in the United States, where they have, as I said, 100 channels. And they record from these 100 channels, and they generate 48 megabits per second. And then if you look at the actual information rate, which is basically the speed at which you can type on this keyboard, then all you get out of this is 5 bits per second, right? So the problem here is that you need a lot of data to generate very little information on the output, right? So this is trying to drink a sip of water from a water hose, right? It's just a lot of water is going to get wasted. And so the idea is, can we do something a bit more clever on this to save power? And why do you want to do that? Because if you save power on a single channel, you can integrate more channels into the same device, right? And hopefully by integrating more and more channels, then we can get to more and more precise control of these processes. So this is a collaboration done with Professor Krishna Shenoy at Stanford University, and at the time, Nireven Chen, who is the co-author with me on the paper, who was a PhD student in Shenoy's lab and now works at Neuralink. And so the pipeline, even in more detail, is you have your signal, your raw signal, and then you basically are interested in just the spikes on this. And so you bandpass filter, and then you look, you do thresholding to find these spikes, and then you do spike sorting, right? So here's a nice view of what spike sorting means, which is you have on a single channel, you have multiple spikes, but they look differently. And so most likely some come from a single neuron, and the others come from a different neuron. And then you use all of those spikes from the different neurons to get your velocity vectors, right? Now, this work starts from the premise that there was considerable amount of literature showing that you do not need to do spike sorting to decode with enough accuracy the velocity vectors. And actually you only get a less than 5% error if you simply do thresholding and bypass the step in which you actually find out whether one spike comes from neuron A or neuron B or neuron C. Now, why that is important is because this can lead to very simple hardware, right? And so now you don't need to transmit the raw waveforms. You just need to transmit zeros and ones, whether there is a spike or not, right? And thresholding is the most popular way of doing compression in neural signals for action potentials. Now, what we did that was different from others, people typically focus on building spike detectors that can be as accurate as possible. And we flip the question around, which is how crappy of a spike detector can we build and still get the velocity vectors to be without errors, right? And so basically, this is how it looks like, right? You have your amplifier, which is basically a bandpass filter with some input for noise. You have an ADC that has some sampling frequency and number of bits. And then you just have a threshold detector, right? And then if the signal is below a threshold or above a threshold, whatever you want to do, then you have a 0 or a 1, right? and then you send that to the decoder, and then you have some metric for the error. In this case, we just look at the R-square error metric here. And so, as we said, this is very popular. We did not by any means invent thresholding as a form of compression. I think this was going on even before I was born. But what we did differently is that we want to define what are the requirements of this thresholder. And so how much noise in the amplifier, what's the bandwidth that we need on the bandpass filter, the sampling frequency, and so on, based on the decoder's outputs. And this is because, so the way we approach this is, unfortunately, there is no closed form because this decoder is highly nonlinear. So you can just find a transfer function and invert it. So we did a data-driven approach that we validated both in humans and in primates. And so we started by using a proxy for the signal quality, which, similar to bit error rate in communication channels, we call spike error rate, where you have your original spike train, right? So here each line represents a spike, so a 1 in your binary data. And then we added random bit flips that resulted in either missed spikes or false spikes, right? And then the rate at which these errors occur is what we define as the spike error rate. And then with these noisy spikes, we try to decode the velocity vectors, and then we look at that and compare it against the velocity vector that we measured in our experiments, because these are experiments done with, let's say in the case of primates, they are actually primates moving a cursor and thinking at the same time about moving. And so we try to estimate the movement, and then we also have the actual movement that they did with their hands, and so we can compare this. In the case of a human, the human cannot move, so there is a bit more details involved that if you want to know more about, I invite you to read the paper. But basically, you get an output without the noise and an output with the noise, and then you try to make the difference, right? And now the very interesting result that we got that got us very excited is that you can be quite liberal in the amount of spike errors that you introduce. And specifically, you want to have your spike error rate just to be slightly smaller than your spike rate. So if you have a spike rate of around 10 to the minus 2, well, you see that when your spike error rate here is on the y-axis, you have your relative performance, and on the x-axis, your spike error rate. So if you go to the left, you go into low noise, high power consumption, and then to the right, you have high noise, low power consumption. And then if you look at 10 minus 2, that's when you start having, let's say, the knee of your drop in performance. But if you go already to 10 minus 3, then basically you have negligible performance drop, right? So you don't have any significant effect. So this was very nice because they said, okay, well, now I can design something that can be very noisy, and higher noise means lower power, right? So that's how we should always think about this. And so the next question was, error rate is not a metric for your amplifier, right? So how to translate this into how much noise you can tolerate, or the bandwidth, or the sampling frequency, and so on. And so basically we did the same analysis for all these metrics, right? So this figure has a lot in it, so I'll drive you through it, right? So on the top left, you have the raw data, and then you can look at figure D, for example, is we explore the bandpass frequency, so the high pass and the low pass pole, F0 and F1, and as you change those, how much change you have in relative performance, which is color coded in black, which was a request from the editor, which I think it's a horrible one, but that's another story. Anyway, you can see that, of course, if you reduce too much your bandwidth, then, which is either you get very low pass filter or very high pass filter, then you have a problem. But you can be quite broadband in these, so you can choose different bandwidths and then still be okay with it. And you can think you can do the same analysis for the number of bits that you want to achieve from your ADC, or the power spectral density of your amplifier, and so on. And then it turns out that you can get very low, very let's say, low performance analog front ends and still get away with it. And so all of this translates, and we show in the paper, to more than 10x power savings. These are extrapolated results. So this is not something we built, but this is comparison with others using the same models. But it allows you to use something like 7 beats and 9 kilosamples per second, which is a lot less than what typically is used in the field, which is around 20 kilosamples per second and 10 beats or so, or even higher. And so what we show here is that if you decide on your compression approach strategy based on the output, you can gain even more, right? And this is a very straightforward thing, because now designing something like this is quite simple, right? In hardware-wise, you just need to know what to design. That's why I call this a low-hanging fruit, because once you do the analysis, then the next step is quite simple. Indeed, I guess there is not much effort into doing this. That so far we haven't done it yet. But I would say that the key is to know how much of the data you can throw away and still get good results at the output. And so basically, this is the conclusion on this part, is that the current motor cortex decoders do not need to spike sort. That's where we start from. And then we found out that these decoders are actually very robust to spike errors. And so then you can do this compression based on the decoder output that allows very large signal distortion, right? In this case, if you look at the input on the left, which is a spike, and the output, which is a 0 or 1, you have, I guess, maximum distortion. But this can translate to very simple hardware while maintaining high performance, right? Now, in parallel with, while we were doing this, actually came out on the same journal. There was another team in Michigan who was looking at spiking band power, where basically they were using a feature that is in the frequency domain as opposed to the time domain, as we were saying. But then they were saying, you don't need to record all the signal and have all the high resolution waveforms. You just want to know what is the bandwidth, so what is the energy in the what they call the spike bandwidth. So what's the power in the what they call the spiking band, which is between 300 hertz and 1 kilohertz. And then they use that to basically study the same paradigm. And the nice thing about this is that this translates again to very simple hardware, right? And so now with that, they have a couple of examples. I think they just came out with a new one, but where they have either analog or digital implementation of this spiking band extraction, which is basically just, you can think about in the analog domain, it's just an integrator with a bandpass filter before and then a threshold, which is a comparator. And then they can build these sub-millimeter square, sub-microwatts implantable modes. So everything from the power delivery, the power management, the recording, the signal processing, and the wireless power transfer, and the wireless data transfer back is implemented in less than a millimeter square and consumes less than a microwatt, which is quite impressive work, if you ask me. So again, knowing what you need will get you very far in terms of your power performance and area performance as well. So that was for the low-hanging fruit. So hopefully the last 20 minutes, I will cover the last part of my talk, which is on, again, an output-driven compression, but in this case for an artificial retina. So we move away from the brain. Now, technically, the retina is part of the central nervous system, so I guess you can still call it a brain-machine interface, but that's just nomenclature, so who cares? You still have neurons, the neurons spike, and you can record from them. I guess that's what matters here. And so what is an artificial retina? It's a device that tries to restore vision by directly stimulating neurons in the retina. And this is effective for treating patients that suffer from macular degeneration or retinitis pigmentosa. So very briefly, what the retina looks like, which is a piece of neural tissue at the back of your eye, responsible for sensing the incoming light. So the incoming light goes to the retina, which is actually transparent, and hits the photoreceptors at the back. The photoreceptors produce some signal that then is processed locally by these output neurons. that transmits all of this through the optic nerve. So all the axons, which are the wires coming out of the neurons, of all the neurons in the retina, form the optic nerve and then goes through various regions in the brain, so that we can see and make sense of the world outside, right? Now, key thing to keep in mind is that these output neurons actually do some local processing, right? What happens in patients that suffer from AMD and RP is that the photoreceptors die, and so the patient is not capable of transducing light into an electrical signal anymore and becomes blind. However, the remaining structure with the output neurons and the connection to the brain are still intact to most extent. And so the goal of an artificial retina is to stimulate directly, bypass these photoreceptors and stimulate the output neurons directly based on an image that we capture from the outside with a camera, right? And so, okay, pretty straightforward. You put electrodes, you stimulate based on the image, and you're done, right? So why is this not done yet? Well, the key thing, the main problem is that the retina is not really a camera, and it's not one pixel equals one neuron, but think of it as many cameras in parallel. So you have different types of output neurons, and here I just show you 4. I'm not going to bother you with the names, but in this case, just keep in mind that they can be as different as on and off type. And so if a neuron receptive field of an on type sees an increase in light, it will start firing, while a neuron of off type will start firing when there is a decrease. And so if you stimulate on the same spot an on and off type, then you will be telling the brain that you have both an increase and a decrease in light on the same spatial location at the same time, which is impossible, and this leads to a very confusing signal that then leads to very poor visual restoration, right? So this means that you need to stimulate with single cell and cell type resolution, which is non-trivial, and it's what we are trying to do. So this is a collaboration with Stanford University, specifically with Ichi Chilnitsky and Boris Moorman over there. And so the approach that we are proposing is a closed-loop artificial retina, where you first record from all the cells. You have a bidirectional neural interface, first records from all the cells, and learns which neurons are where. And so starts building a dictionary of all the available neurons we can record from. After that, you want to know what are the accessible neurons. So if I want to stimulate neuron A and not neuron B, okay, how much current do I need to pass on each electrode, right? And this is what we call the dictionary calibration, where you stimulate and record at the same time. And only after you build this dictionary, you can do what we said before, which is you have an image, you know which neurons you want to activate, you take your dictionary elements to activate those, and then you have your runtime, right? Now, today we're just going to be focusing on the recording part and why we need compression in here. If you want to go and have a look at more details, actually, EJ and I wrote a book chapter on CHIPS 2030 some time ago that basically describes all the basis of the project. So, single cell resolution, right? So now we really need to understand whether a spike comes from one neuron or not. And this is the process of spike sorting that I was telling you before, which is basically just looking at the different waveforms and then try to do classification to see whether they all come from a different place, right? And the way you do this is you just plot them in a low dimensionality space where you just use the features of the signal to have your axis, and then you just do some clustering, right? So we don't need to go into the details of this. What you need to know is that you cannot do spike thresholding because you need to look at the waveform. That's how you can do spike sorting. And not only that, but you need to look at the whole array at the same time, because you want to record from all channels simultaneously, because you need the spatial progression and the correlation of the signal throughout the array. And I'll show you an example later on for the cell type classification. So basically, you need features like how fast a spike propagates through your recording array to classify between different cell types, or how large is the footprint of a cell, because you can record the same cell from different electrodes. So how many electrodes can you record the cell from? And this is what we call the footprint of the cell on the array. to differentiate different cell types and so on. So all of these to justify that you need to record the waveforms, and you need to record from all the channels at the same time. Okay, so can we do it? And can we do that for an implantable system that has, in this case, for various reasons, a power limitation of around 10 milliwatts that comes from basically how much power you can dissipate in the brain or in the retina, in this case, without burning the tissue, given the size of your implant, right? And so you want to have here your front end that has its low noise with enough bandwidth and enough resolution in your ADC. And now we don't go back to the numbers I showed you before, because again, we want to have the whole waveform, right? And so you look at state of the art and you look at let's say physical limitations, and then you say, okay, this is not too bad, right? We are talking about, maybe, let's say this is quite optimistic, 50 nanowatts, but maybe 100, 200. There are examples out there with 200 nanowatts. And so you can say, let me go on crazy and just integrate 100,000 channels. That's my goal, in the next 10 years to get here, right? So what is going to be the main limitation, right? And it turns out that maybe the recording scalability doesn't seem to be a big issue, right? But when you look at the system, the aggregated system data rates, now you're talking about 16 gigabits per second that you need to transmit, right? If you have a very optimistic 10 picojoule per bit in your transmitter, now you're burning more than an order of magnitude higher power than what you actually have available, right? And so again, here, I guess, no surprise, we need to do on-chip compression. We want to do it near the sensor. But the problem here is even harder than that. It's not just, okay, let me put my favorite DSP here that does compression from whatever I showed you before and call it a day. Because even the cost, when you have this many channels, of moving from your array to whatever DSP that you have outside the array inside the same chip, right? So let's say a 1 millimeter wire and to store into an SRAM cache of 100 kilobytes, right? These are not crazy numbers, but the cost of this when you want to do gigabits per second already goes above your power budget, right? So you're here talking almost 20 milliwatts just for moving the data, nothing else. And we still haven't even accounted for the cost of actually compressing. So this is kind of a problematic situation to be in. And so, we are kind of up against the wall. What can we do? Well, we apply the same approach as before, which is looking at the data and seeing, looking at the signal, characteristics in the signal, and the processing we need to do with it, and see what we can do about it, right? And so this is what we looked at to begin with. So on the top left, you see a single spike, how it looks like. And then on the bottom, each circle is actually one of the electrodes, and the size is what we're recording. And so here you see these spikes propagating, as I was telling you before, through our array, right? So these are the footprints that we see. And this is actually real-time spike proliferation of isolated primate retina that we recorded in the lab. And so one thing you see is that the data is quite sparse. So you don't have spikes all the time, and it's sparse both in space and in time. And so you have a compression opportunity for sure. Nicely, this is sparse already in the time domain, which means we don't need to do any transformation, which makes it easier for us. And then also, the other thing is that when you look at the actual data, information is only encoded in what we call the blue samples here, which are belonging to the spike. And when there is no spike, the signal is around the baseline. And we call that red samples in here, and we just say that they carry no information. Furthermore, because we care only about the output, we don't need to record the spikes perfectly. We just want them to be good enough to distinguish spikes coming from different cells, so that we can build these beautiful mosaics you see on the left, where basically every circle is our estimation of where the cell is, right? So as long as we can reconstruct this, we don't care if the spike is all, distorted. Who cares? As long as I can do what I want to do. So these are the three main things, right? Signal is sparse in space and time. I only care about blue samples, and I do not care about the blue samples perfectly, as long as I can reconstruct my my mosaics. So the question now is, how can we toss out the red samples without spending much energy? And remember that we want to avoid spending energy on full digitization of the data, moving the data around, and of course compute, right? And so before telling you the exact algorithm we implemented, let me just start from the actual analog to digital converter architecture we use, which what we chose was a ramp ADC. So what a ramp ADC does is basically it has the inputs that you sample, and then you compare it against a single ramp that keeps going up, a staircase in this case. And then when the ramp crosses the input, then the comparator will send out a pulse. And if you know at which time the pulse arrived, then you know the amplitude of the input, which is your basically analog to digital conversion. And so it's a voltage to time to digital conversion, right? So if the input is smaller, then the pulse will arrive sooner. If the input is higher, then the pulse will arrive later. Now, why using a ramp ADC and not a SAR ADC, for example? Right, so a ramp ADC has a linear search, and a SAR ADC has a binary search, which is more efficient. The nice thing about ramp ADCs in array problems, and this is why it's used often in imagers, is that no matter the input, the ramp will always, the DAC that implements the ramp will always do the same thing. While in a SAR ADC, the steps that the DAC takes depend on the input, right? And so this allows us in the ramp ADC to share this DAC among all the different channels and just put a tiny comparator inside the pixel, right? And so this is why we went for the ramp. However, we still need to read out these pulses. And if we want to read them out from each pixel as a memory with let's say a raw-based readout, then we'll be burning a lot of power, right? So instead, let me play a trick that I'll justify in a second that gives the name to the actual compression algorithm that is called wired OR compression. And we have published a T-Biocast paper a few years ago, and then you can read more about it. But basically, the idea is very simple. You have this voltage to pulse conversion on the pixel that we just explained how we build. And then instead of reading out each individual pixel, I connect the rows and I connect the pixels to one row and one column in a wired OR fashion, right? And so, for example, if at t = 4, all these pixels fire, then at that time, I will have this column and this column and this column and this row, basically all the columns and all the row firing up, right? And then I only look at the row and columns at the step, the ramp step to decode the output, right? Now, immediately you see how this is a problem, because in this case of t = 4, it is impossible to decode, right? And so we call this a collision, and a collision happens when you have the same sample for two channels or more at the same time. And so from the outside, here's an example of just two, and you can see how this is impossible to decode, because if you only look at the rows and the columns, then really it could have been the two that actually fired, or these other two that we call ghosts for fun, or all four of them at the same time. And so you cannot tell as a result. Our decoding strategy is just to throw away this data, and we say, well, this is data we don't care about. And sometimes we have a no collision, where you have a unique channel that has a unique value, and then from the outside, basically what you get is the X and Y coordinates, right? The row and column address of this channel. And so these ones are the ones that we store and process. Now, again, one could ask you, well, Dante, okay, this is very nice, and it leads to very simple hardware, but you are throwing away a lot of data. How can you ensure that the no collisions are the data you want to store, right? And so let me first give you an intuition, and then we will justify this by looking at the data. And this is how it looks like, right? So if you have your spike and you look at how these samples on the spike map to your ramp, right? You will see that most of the blue ones map to unique places in the ramp, and all the reds kind of like go to the baseline in the middle, right? And so these baseline samples will always map to the center of the ramp, will generate massive collisions when the ramp is crossing its center point, and will not be able to be decoded, but that's actually what we want. And the spike samples will map to hopefully unique locations in the ramp, which will allow for decoding, right? So mathematical way of looking at this is by simply looking at the probability of spike samples having, this is the probability of the channels with the same digital code at the same time, right? And so everything with more than x more than 1 in here is a collision. And so when you look, this is from real data from the retina, this is actually kind of nice because spike samples are most likely no collisions and baseline samples are most likely collisions. However, you can still get some spike samples to generate collisions. So this is actually lossy compression. And so the question is, well, if the compression is lossy and depends on the statistics of the input signal, how do we know if it's not too lossy, right? So if the reconstructed signal is good enough. And so again, we go back to a data-driven approach. And in this case, having a collaboration with the Chichinitsky lab has been great because they have the equivalent. I think I never found anything more closer to big data in biology than what they have. So they've been recording from the retina for the past 20 years and have something like 50 billion spikes recorded. And so we can apply this compression to all this data and see whether we can still record these mosaics before. And if we can, then that should be good enough for us, right? And so this is what we show originally in our T-Baricos paper, and now we just came out with a different paper in 2022, validating this also outside of the retina, that will come out in Biocast in next month, actually. So if you are there, I suggest you check out the presentation by Pumiao Yan. And so this is how it looks like, right? So you have your original blue data, and then you only have a few collision-free samples. And the reconstructed data is actually a bit noisy, and you miss a few samples here and there, and your spike doesn't look perfectly. But it allows you to reconstruct these mosaics with more than 95% accuracy while achieving around 40x compression. We validated this across 3 different primate retinas. And basically, that's it. So this is, in our case, this is good enough for us, but the approach is always to go data-driven. And so the nice thing about this is that it leads to a very simple architecture where all you need to put inside the pixel, okay, you have your conditioning, whatever amplifier you have, but that you always need. And then you need a comparator and 2 switches for your wire door, and that's it. And then everything else can go outside, and this allows you to do very simple, small and low power pixels. And so this is still not published, so we are measuring right now, but it's a picture of the implementation in this case, where we have all the electronics for amplification, ADC, and compression inside each of these tiny pixels in here in the center. And the pitch is 35 micrometers, and we are burning around 300 nanowatts per channel. And this is for amplification, digitization, compression, and data movement. So it's all in one. So we are still, a bit far away from the 50 nanowatts per channel I said at the beginning, but I guess this would be state of the art the moment we get to publish it. And so just to summarize, because I think I'm running out of time. So goal-driven compression, I think hopefully by now you agree with me, it's the way to go when you are very limited in the power and area budget. And so in this second part with this wire auto compression, I show that you still can do compression when you need to spike sort. So thresholding is not the only way. And again, spike sorters are robust to spike waveform distortion. It turns out that typically a lot of the processing you do on the data is actually quite robust to you doing a lousy job on the recording side. So that's good for us, I guess. And in this case, again, if you do your compression based on the output of the spike sorter, then you can allow for larger signal distortion, and all of this can translate to very simple hardware, which in this case we build and hopefully will be publishing soon. So just want to acknowledge collaborators and funding for this. Here are for your reference. I guess I'll go through quickly. Hopefully, if this is recorded, then you can go back. But if you need any paper, just you can send me an email. And I guess that's all from me. So thank you for listening. And if you have any questions, I'm here.
So thank you very much, Dante, for the really interesting talk. I think I'm going to start with the question as they trickle in the chat. So I think it's very clear, like from your approach, that you really need strong partnerships, right, to perform this type of research. And they're interdisciplinary in nature. So what are some of the challenges you came across when starting this?
Many. Well, first of all, yes, absolutely. You need strong partnerships. And this takes a lot of time to build. And you need to learn. I think the first challenge for me was learn to communicate with these partners, because when I started working on this during my postdoc. So I barely knew what a neuron was, right? And so you need to take the time to educate yourself. You will never become an expert. You need to be an expert in your own field, right? And I am an IC designer, and this is this is what I do best. But you need to take the time to educate yourself in the field in which you want to be making an impact, right? In this case, for me, it's neural engineering. So that you can ask the right questions to your partners, and you can understand whether your partner knows something about it or not to some degree. That's one thing. And then the other is, like in any collaboration, you just need to be a good collaborator yourself, right? Just be a nice person and people want to work with you. I guess that's always true. And allocate time for building a good team, because it takes considerable amount of time. So if you have an idea like I would like to do this for, I don't know, an implant on depression, before you put together the right team, it might take you a whole year, right? So but then once you do, then you can do these amazing things. So I think it's worth it.
Okay, thank you. We have a couple of questions trickling into the chat. So the first one is from Professor Sergio Bumpy from Federal University of Rio Grande do Sul in Brazil. If you're placing questions in the chat, please state also your affiliation. Thank you. So a question from Professor Bumpy is, the retina chip is interfaced inside the eye to tissue. And how is this physical interface made?
Okay, so there are different approaches, but ultimately is some sort of electrode. And so the key is that if you want to achieve single cell resolution, you really need to have the electrode very close to the tissue. And by very close, to give you a rough number, if the distance is 20 micrometers, the stimulation threshold doubles. So this is something to give, and it's exponential, right? So this is the how close you need to be. Now, one thing that is nice about the retina is mostly 2D structure. And so you don't necessarily need penetrating electrodes. However, in our case, we are studying these nano pillars that we 3D print directly on top of the chip using nanoscribble, which is a very fancy machine that allows you to 3D printing at nanoscales effectively. And this is to ensure also a bit of friction such that the chip, once you push it against the retina, doesn't move, right? But typically you have electrodes, and most common materials are platinum black or Syrof, which is sputter iridium oxide film, and things like that. You have some organic approaches like P.PSS, but they haven't been they haven't shown yet enough stability over time. But the interface is made of electrodes. And what we do, the moment you want to have, thousands of channels, the issue is that if you have your electrodes that are not monolithically integrated with your chip, then you need to have a 1,000 channel wire inside the retina that is flexible and connects. Connections are where things break typically, and so our approach is to build these pixels that are matched to the electrode pitch. That's why we want to do 30 micron, 35 micron pitch on the pixel, because then we can put the electrode directly on top, and then everything becomes a single device, and then we don't need to have a wire inside, which is a big problem. Hopefully that answers the question.
Yes, thank you. We have another question from Sid Abubakar, and sorry if I'm mispronouncing your name. Is there any open source database available for the spikes data for brain or retina?
Yes, absolutely. There is plenty. The data I show is not publicly available. So these are, let's say, proprietary databases from in the retina from the Chichilinsky lab and in the brain from the Chenoy lab. But there is plenty of available, both real and artificial databases, and they both have their pros and cons. So you can start by looking into those, right? Here's another, going back, Oscar, to your question on collaboration, right? This is another thing that it's a huge benefit of having great collaborators is access to data, right? Because yes, there are open source databases, but maybe they're not as well maintained, or you don't have the person who collected the data explain to you the data as well, right? So I could go into the lab and see as they were recording and asking, okay, well, what's the gain, and and things like that. So unfortunately, that I have is well, it's not mine and it's not available publicly, but there is out there stuff for you to get started if you need to.
Okay, thank you. I have another question that's related a bit more to circuit design, though. It seems like some industries or some applications require circuit designers to know a lot more about advanced DSP, about algorithms, and in your case, like even for biology, right, and biological systems. But it's still a transition, right? Going from being a hardcore circuit designer, which is somebody that can learn about these other fields. Do you still see a role for advanced circuit design, at least in your current research areas?
That's a very difficult question. So can I get a lot of people upset? But I think there is still a role for advancing circuit design as a field in itself, right? So how is the most efficient way of converting data and these generic purpose things? I think there is still a role, and well, I mean, there is a very mature industry that benefits from that. Given that said, the way I see circuit design and the way I get excited about it is as an enabler to things that we cannot do. And now, because circuit design is very complicated, to tape out a chip with like a million transistors in there, there is no way you can do that on the side, right? I'm an expert in biology and I learn how to design a chip and then I do that, right? So you need to be an expert in it, and that's where you come from. And I think that you have room for advancing circuits, specific, maybe a circuit trick here and there, but it shouldn't be the main focus, right? The main focus, in my opinion, you get the biggest gains at the system. And so for that, you need, the truth is that you need all of it, right? You need to know about circuits, so you know how to reduce the power, what makes you win in the end. You need to know about signal processing because that's the data ultimately is analog to information that you're trying to achieve, and you need to know about the biology. But you still have a few tricks here and there that you need to play, right? For example, this pixel that we designed, 35 microns. So how to fit an amplifier with a low pass, high pass frequency of 100 hertz in a 35 micron, in which a third of that pixel is actually ESD protection. So how to do that requires advanced circuits that make me proud of. But doing it just for the fun of doing it, I think it's not as relevant as having the whole application to justify it. So I would say 30 years from now, is someone gonna get a PhD just out of designing an amplifier alone? Probably not. But you can. I think there's still a role in advancing circuit design for driving new applications.
Okay,
but
thank you.
That was a hard question. I don't know if I completely happy with my answer, but I've been thinking about it for a long time.
Yeah,
More.
Yeah, the purpose is really just to have the discussion going, and I'm sure there's people watching this talk that are interested in these sort of questions these days. We have another question from Professor Sergio Bumpi, and he's saying that the MEMS part of the implantable retina system is a great challenge, and he's asking if for a 1,000 up to 10,000 channels, if there's a good known solution in the future.
Yeah, so I agree. This is a critical challenge. So let's say that we get to 100 on a chip that has 100,000 channels. Well, how to connect that to the electrodes? So looking at out there. So this is a bit nearby field, but not something I'm an expert on. But I would say there are three interesting approaches I've seen outside that I can point to. Right, one is the one that we are doing, which is basically this 3D printing directly on top of the chip, and then you need to 3D print microstructures so that you can get enough height so that they can push the chip against whatever you want, right? The other is what Neuralink is doing with this sewing machine with these needles, which is, if you ever seen a video of that, it's just blind mowing. They literally are just sewing threads with 32 electrodes into the brain, and the machine directly avoids any blood vessels in real time. So while the brain is moving, so pretty crazy stuff. They still have the problem of connectivity, and indeed, if you see, then the chip is quite large. And then I would say the third approach is the bundle of micro wires developed by Nick Meloche at Stanford University, which is literally a bundle of micro wires that then is pushed against the chip, and that's kind of like a bump bonding onto the chip itself. And then on the other side, you have the electrodes altogether. And they got already to, I think they demonstrated 10,000 channels or something like that. And so those are the approaches I know of. But basically, in general, you want to look at what Neuralink and Paradromics are doing in the let's call it industry, the very, very young industry of brain-machine interfaces. Very exciting one, though.
Okay, thank you once again. At the moment, we don't have any other questions on the chat. So I think in the name of everyone that... I'm sorry, we do have an extra question coming in. So from Professor Paulo Diaguirre. And he's asking which technology is being used to design these circuits.
So in this case, we use TSMC 28 nanometers. Now, if you want to put in all this level of processing and have these pixels be small, then you want to go to something scaled, right? And the trade-off here is analog performance and high voltage compliance versus scalability, right? So I think you cannot go to like FinFET probably, but you don't want to go to 180 nanometers, which what typically is used in these bioimplants. So 28 was what we found to be a good compromise. You still have 1.8 volt devices, and if you want to put processing in there, you get quite enough for the back. So that's good. That's what we used.
Okay. So I think with that, we can conclude. Thank you very much for the very interesting talk, Dante, and thanks for being with us.
Yeah, thank you. Thank you for having me. This was fun.